The ABO blood group system. Our objectives are to describe the reciprocal relationship between ABO antigens and antibodies, describe the immunoglobulin class of ABO antibodies, describe the effects of age on the production of ABO isoagglutinins, predict the ABO genotype and phenotype in offspring from various ABO matings, explain the formation of H, A, and B antigens on the red blood cell surface from the precursor substances to the immunodominant sugars. Describe the relative activity of the H antigen in the ABO blood groups. Objectives. List the immunodominant sugars associated with H, A, and B, and SE. Describe the formation of H, A, and B soluble substances. Describe the qualitative and quantitative differences between A1 and A2 phenotypes. Describe the reactivity of Ulex europus with various ABO groups. Describe the characteristics of the Bombay phenotype. Explain the effect of disease on the expression of ABH antigens and antibodies. Interpret the results of ABO testing and resolve discrepancies, and list the common sources of technical errors associated with ABO discrepancies. The ABO blood group system was the first system of human blood groups that were discovered. Carl Landsteiner discovered the A, B, and O types in 1901, but he didn't discover the group AB until 1902. He did this by mixing the cells and plasma from different people and seeing the reactions. This is the single most important test performed in the blood bank, but it took another man to suggest in 1907 that cross-matching should be done between donors and recipients to ensure compatibility, and these were direct artery to vein transfusions. We didn't even have anticoagulants for collecting blood until 1914, and the use was not accepted for more than 10 years. The first blood bank wasn't established until 1932 in Leningrad Hospital. The United States didn't get its first blood bank until 1937 at the Cook County Hospital in Chicago. The ABO blood group system consists of 3 genes, which are A, B, and O. These genes produce 2 antigens, which are A and B. There are 3 antibodies, which are anti-A, anti-B, and anti-A, B. When the anti-A, B is high titer, it is responsible for fetal-maternal ABO incompatibility, where the type A, AB, or B cells of a fetus can be coated with the antibody produced by a type O mother. This is the most common and least severe form of hemolytic disease of the fetus and newborn. The ABO gene is located on chromosome 9. The A and B genes are considered codominant, and the O gene is an amorph that does not produce a gene product. So when thinking about people's types, you would treat the O gene the same way as you would recessive. There are 4 major blood groups, which are A, B, AB, and O. If you remember these facts, it will help you to figure out many ABO group questions. There is a difference between phenotypes and genotypes. Genotypes are the genes inherited from both parents, and the phenotype in blood bank is the blood type, or the antigens expressed on the red blood cells. So when we talk about phenotype or our blood type, we mean A, type B, type O, or type AB. For a person who is type A, or for a person who is type A, they could have 1 or 2 genotypes. They could be either AA or AO. For type B, the genotypes are BB or BO. If you use a Punnett square, you could see that it's possible for a type A mother and a type B father to have children of all 4 blood types. That is, if they carry one O gene. This slide shows you a summary of the ABO blood group phenotypes. The 4 phenotypes are listed. Next, we see the antigens that are on the red blood cells and the antibodies produced by people of each phenotype. For group O individuals, their cells will lack the antigens of the ABO group, and the plasma will have anti-A and anti-B. Group A individuals have A antigens and produce anti-B antibodies. Group B individuals have B antigens and produce anti-A antibodies. Group AB individuals have both A and B antigens, and they do not produce antibodies to the ABO antigens. Notice the reciprocal relationship. If you have an antigen, you don't make the antibody. If you don't have the antigen, you make the antibody. When we look at the frequency of ABO groups in the US, you will notice that there are differences among the different races. But overall, type O is the most common. Type A is the next common, then type B, and last type AB is the least common. These frequencies will not be the same in other countries. For example, when I was stationed in Iraq, the Iraqi people showed a significant higher frequency of type B and AB than was normally seen among the US military members. You can see that Asian people have a frequency of B that is double that of the white Americans. The black Americans are nearly twice as likely to have type B than white Americans. Frequencies of other blood group antigens also differ widely. This can be important to know if there is a patient who has antibodies, and you may have a better chance of finding a match in a certain group of people. Although the antibodies are naturally occurring, they are not detected at birth. Therefore, we do not test newborn back types for these antibodies. The anti-A and anti-B become detectable by 3 to 6 months of age. The ABO antibodies decline with age and may become undetectable by conventional methods in elderly patients. In blood bank, we have forward grouping or front type and reverse grouping or back type. The front type is the cell type and tests the unknown red cells of the patient with known antisera to determine the ABO antigens. The red blood cells are tested with anti-A and anti-B reagents. The back type is the serum type, and we test the patient's serum or plasma with known reagent A cells and B cells to detect the antibodies that are present. We must then compare the front type and back type to ensure that they match before reporting the blood type. This table shows the ABO forward grouping and the expected results for each. If anti-A plus the patient's cells is positive, then A is part of their type. If anti-B plus the patient's cell is positive, then B is part of their type. Notice that we use 0 to indicate the negative reaction, and that the positive reactions have a grading of their strength. ABO front types are normally 4 plus, or the strongest reaction. People who are type O have 2 negative reactions, and type AB people have 2 positive reactions. Notice that O and AB have opposite reactions. Notice that A and B have opposite reactions. The ABO antibodies are generally IgM, but they can be IgA and IgG class also. These are considered naturally occurring because we do not have to be exposed to red blood cells to generate the antibodies. These are the only naturally occurring expected antibodies that we should find. All others are unexpected. They react at room temperature after centrifugation, or we call it immediate spin reaction. These antibodies react strongly and can bind complement. The anti-A and anti-B that are IgM do not cross the placenta, but IgG anti-A, B made by type O mothers can cross the placenta. This table shows you the reverse grouping results of the 4 types. Notice that if you hold these next to the front type, that the results look reversed. A person who is type O has plasma antibodies that will agglutinate both A and B reagent cells. Type A individuals have antibodies that agglutinate type B cells, and type B individuals have plasma antibodies that will agglutinate type A cells. Type AB plasma does not agglutinate type A or type B cells. Remember that ABO testing shows the inverse reciprocal relationship between the forward and reverse types. The antibodies are naturally occurring and are expected to be present. The theory is that exposure to our natural bacteria or pollen causes us to create these antibodies, but no one has discovered what the cause is. The H blood group antigen is required for the ABO blood group antigens to be created. The H blood group system has two genes, capital H and lowercase h. The system has just one antigen represented by a capital H. The lowercase h gene is an amorph and does not create a gene product. A person homozygous for the amorph gene, lowercase h, what is known as Bombay phenotype, these people make an anti-H, capital H, which will react with all other types that are not Bombay. The gene for the H group is found on chromosome 19. These genes are not linked to the ABO genes and do follow Mendelian law of independent assortment. They are not inherited together. The Bombay phenotype is extremely rare, and they do have naturally occurring anti-H, making it very difficult to find compatible blood for them. The rare donor registry must be consulted, or frozen blood from a donor with Bombay phenotype must be thawed for transfusion. Inheritance of at least one capital H gene causes the production of the enzyme alpha-2 L-fucosyltransferase, which will transfer an L-fucose to the terminal galactose of a precursor substance or paraglobicide. The H antigen is the precursor molecule on which A and B antigens are formed. More than 99.99% of the population has this antigen. When a person is either homozygous or heterozygous for the capital H gene, they will generate the gene product, which is a fucosyltransferase. This fucosyltransferase will add the L-fucose to the precursor substance to create an H antigen. The immunodominant sugar responsible for the H specificity is fucose. This is the only antigen formed by people who are type O. Therefore, this is the immunodominant sugar for type O people. The A and B genes code for the production of transferases that add the immunodominant sugar to the H precursor molecule. The A gene codes for the production of N-acetyl galactosamine transferase, which adds N-acetyl galactosamine to the H substance on the red blood cell surface, creating an A antigen. The enzyme is the most efficient at converting H antigens to the A antigens, so people with an A gene have fewer H antigens than those with a B gene. The B gene codes for the production of D-galactosamine transferase, which adds D-galactose to the H substance on the red blood cells to create a B antigen. The immunodominant sugar for type B is D-galactose. When the A and B genes are both present, the B transferase competes more efficiently for H. So a type AB person has slightly more B than A antigens. The H substance must be present on the cell surface in order for A or B antigens to be expressed. A person with a normal A and B genes, but who has the Bombay phenotype, will not produce the A and B antigens, but will pass these on to these genes will still be passed on to their children. This slide shows the formation of the A and B antigens. Notice that a single sugar is the difference between the blood types. The gene product is a transferase. It is not the antigen. A person who is type AB does not make an AB antigen. They have both A and B antigens separate on their red blood cells. There are two types of precursor substance that can be converted to H substance and the A or B antigens. The type 2 is found on the surface of red blood cells, and the N-acetyl galactosamine has a beta-1,4 linkage. Type 1 are soluble and are found in plasma and body fluids. The terminal galactose on type 1 precursor substance is attached to the N-acetyl galactosamine in a beta-1,3 linkage. The amount of detectable H found on red blood cells varies by starting from greatest to least. We have O, A2, B, A2B, A1, and then A1B. The O gene does not produce a transferase at all, So the H antigens on the red cells is the highest. So they have the most. The A gene produces the highest concentration of transferase, which leads to conversion of practically all H antigens for A1 individuals. So in A1B people, you have the best transferases working to convert the H antigens, so they have the least amount of H left. This is a good chart to memorize. It has the gene product, the immunodominant sugar, and the antigens. So if we start with H, the product of translation is an enzyme, alpha-2 fucosyltransferase. The immunodominant sugar is L-fucose, and it's the H antigen. The A gene produces alpha-3 acetylgalactosamine and the immunodominant sugar is N-acetyl-D-galactosamine. This is the A antigen. The B gene produces alpha-3 galactosyltransferase, so the immunodominant sugar is D-galactose and the antigen is B. The O produces no enzyme, so the immunodominant sugar is the L-fucose, and this is the H antigen. For AB, they produce the same two enzymes produced by the A and the B gene, and they have both um, N-acetyl D-galactosamine and D-galactose on their cells, so these are the A and B antigens. Another system that works with the ABO and H system is the secretor system. This is a separate gene that has two genes, capital SE and lowercase se, and this regulates the formation of H antigens and subsequent A and B antigens in secretions. So this would work on the type 1 precursor substance. The secretor A, B, and H antigens are glycoproteins. The A, B, H antigens on cells are glycolipids. The capital SE gene does not affect the formation of A, B, or H antigens on the red blood cells. One copy of the capital SE gene must be present for soluble substances to be secreted. Again, the secretor's gene, a system has two genes, capital SE and lowercase se. This gene is found on chromosome 19 and are linked to the genes of the H and LE or Lewis loci. 78% of the random U.S. population are secretors with either homozygous or heterozygous for the capital SE. 22% are non-secretors, homozygous for lowercase se. For group A, there are subgroups that are slightly different and can have some incompatibility. There are different transferases, one made by the A1 gene and one made by the A2 gene. Approximately 80% of all group A individuals are A1 and the other 20% are A2. Other subgroups of A exist but are rare. There are both quantitative and qualitative differences between A1 and A2. Most of the time, these cells will give a front and back type that looks the same. Occasionally, some A2 samples will have a discrepancy between the front and the back type, requiring extra work to resolve the discrepancy. When we have the discrepancy, we must prove that the person is A2, and this is the reason for the difference. When this happens, the front type will look like A, but there will be an anti-A in the back type. To differentiate an A1 for an A2, we're going to use an anti-A reagent and an anti-A1 lectin that will react only with one type. So both A1 and A2 will react with anti-A that we normally use for typing, but if we use anti-A1 lectin, it will react only with A1 cells, but it will be negative for A2 cells. A lectin is any class of protein, usually of plant origin, which binds specifically to certain sugars and causes hemagglutination of particular cell types. The lectin with anti-A1 specificity is known as dolichos biflorus. Red cells from group A1 individuals will agglutinate with this lectin, and red cells from group A2 will not. Group A2 individuals may form an anti-A1, which is considered unexpected, but it is not usually clinically significant. However, instead of transfusing these patients with type A, we will use type O. There are other weaker subgroups which exist, but they are very rare. They have a decreased amount of A antigen sites and an increase of H antigens. These are less than 1% of the group A individuals. Weak subgroups of A may form naturally occurring anti-A1, and they may type as type O. Weak B subgroups are much rarer than those in group A. If a subgroup is not recognized for what it is, the sample is usually typed as O. If this was a recipient, this would not be a problem because they would just get type O and that would be compatible. In the case of a donor, if a unit of blood is mistyped, and so it would be A, but it would be labeled as O, the anti-A, B and A recipient could attach to these A antigens and cause a transfusion reaction. A person who has the Bombay phenotype lacks H, A, and B antigens. This would type as O, but it would be incompatible with all group O cells in a crossmatch. If we used anti-H lectin called Ulix europus, the test would be negative. There will be anti-A, anti-B, anti-A, B, and anti-H found in the patient's plasma. The patient must receive blood from a Bombay donor. The rare donor registry will have a list of suitable donors. There may be frozen blood that can be thawed for the patient. If a surgery is planned, autologous blood can be donated. These are what the normal ABO results should look like for each blood type. You should have this memorized and be able to know if there is a problem with the results. When we do a front type, we're going to mix the patient's cells with anti-A and anti-B, centrifuge, and observe for agglutination. A front type normally also includes anti-D, D control serum, but this is the RH type, and we'll cover that in another lecture. The reverse type combines reagent red cells, A cells, and B cells with the patient's plasma or serum. Then it's centrifuged and observed for agglutination. An ABO discrepancy occurs when the expected positive and negative reactions are not observed in the front forward typing or in the reverse typing. For example, the expected forward typing does not match the expected reverse type. A discrepancy also occurs if the ABO type does not match the historical record of the patient's type, unless there is a record of the patient receiving a marrow transplant of a different blood type. The first thing you would check is the labels on the red cells and plasma, and then you would repeat the test to see if, as long as you have the proper samples in the first place. You will need to remember the different types of discrepancies and their causes. Group 1 discrepancies are caused when there is a missing verse type due to weak or missing antibodies. This can occur naturally in newborns or the elderly. This can also occur if someone is immune suppressed due to drugs, HIV/AIDS, or a disease state. A more unusual case would be if the person had two cell populations, their own plus those from a marrow transplant. The person will not create antibodies that react with either population. Reviewing the patient history is important in these cases. To resolve a type 1 ABO discrepancy, you will incubate the patient's serum or plasma with A1 and B cells for 15 to 30 minutes at room temperature. If this doesn't resolve it, you can also incubate the patient's serum or plasma with A1, B, and O cells at 4 degrees Celsius, that's the refrigerator, for 15 to 30 minutes, with the O cells acting as a control cell. This should be negative. ABO antibodies should react more strongly with the additional incubation time or a decreased temperature. Group 2 discrepancies can occur when the forward and reverse typing seem altered due to missing antigens in the front type. This can occur with weak subgroups of A or B, when a person has a disease state such as leukemia, or when there is an acquired B associated with intestinal obstruction or malignancy of the stomach or intestines. Acquired B means the molecules attach to the cell and they react like a B antigen. To resolve the group 2 discrepancies, you may incubate the patient's cells with antisera for up to 30 minutes at room temperature. So for group 1, we were incubating the back type, and for group 2, we were incubating the front type. If it's still negative, incubate the cells in antisera at 4 degrees Celsius for 15 to 30 minutes, including a type O screening cell and autocontrol. An autocontrol is when you mix the patient's cell with the patient's plasma, which should not agglutinate. Group 3 discrepancies are due to proteins or plasma abnormalities that cause pseudoagglutination called rouleau. The cells will look like stacked coins under the microscope. The cause of this can be increased levels of globulin, such as what is seen in multiple myeloma or Waldenstrom's macroglobulinemia. If the patient is being given plasma expanders, or if there is Wharton's jelly that has not been adequately washed from cord blood cells. To resolve the type 3 discrepancies, the patient's cells should be washed additional times to remove the plasma. A saline replacement will disperse rouleau, but not true agglutination. Washing cord blood cells 6 to 8 times with saline to get rid of the Wharton's jelly will resolve that issue. A group 4 discrepancy might be polyagglutination, cold reactive antibodies, or unexpected ABO isoagglutinins. A cold reactive antibody could be either an autoantibody or an alloantibody. An alloantibody could be reacting with an antigen other than A or B on the red cells, and an anti-M would be a good example of this. To resolve a group 4 discrepancy, such as polyagglutination, we would use a monoclonal antibody. To resolve cold reactive antibodies, we can remove using autoabsorption if a patient has an autoantibody and they have not been recently transfused. Group 4, unexpected ABO isoagglutinins. An unexpected ABO isoagglutinin would most likely be found in A2 or A2B individuals who produce anti-A1. More uncommon would be a person who is A1 or A1B to produce anti-H, since they convert all of their own H antigens. They would think of that as foreign. You can have unexpected alloantibodies in the patient's serum other than ABO agglutinins, which may cause a discrepancy in the reverse typing. These IgM antibodies can be naturally occurring and they react at room temperature. If an individual's serum contains, say, an anti-M, a discrepancy in the reverse typing may occur if the reverse typing cells possess the M antigen. To resolve this issue, you may use A or B cells that lack the M antigen to do the back typing. The phenomenon of cis-AB is an interesting rare phenomenon. A parent donates both A and B genes on one chromosome. This is thought of to be due to crossover between chromosomes during gamete formation. The A and B antigens are weak in this case. You can see that we have some Punnett squares down here, and the first one would be what it would look like with a cis-AB. So say the person had AB on one gene, and then they had this one right here, AB on one, and then O on the other. And so when you crossed it, you'd have an AB gene from one parent and O from the other, instead of what would normally happen, which is over here, where you would have the A and B inherited separately. So it's an unusual and interesting case. Thank you for listening.